You reach for an apple at the grocery store without a second thought. You toss spinach into your salad, sprinkle herbs on pasta, or bite into a crisp carrot. Every day, billions of people consume thousands of different plant species, trusting they will nourish rather than harm. Yet this casual confidence rests on one of humanity's oldest and most hard-won bodies of knowledge, the ability to distinguish safe, edible plants from those that could kill. How did our ancient ancestors, with no laboratories, no written records, and no scientific method, figure out which plants were safe to eat? For millions of years, our early ancestors lived as opportunistic foragers. Plants were abundant, but carried hidden risks. Compounds that could cause immediate discomfort, delayed illness, or death. In a world without reliable knowledge, a single mistaken choice could prove devastating, especially for the young or vulnerable. Survival depended on learning what could be eaten and what must be avoided. Picture a small group moving through a woodland edge long ago. A child reaches toward bright berries. An adult gently guides the hand away. No one yet possesses a complete catalog of safe foods, but painful experiences have taught caution. One possibility is that the group had witnessed illness after similar berries were eaten weeks earlier. The memory lingered in stories and warnings. This kind of direct experience and social learning formed the fragile foundation of early plant knowledge. Groups that repeatedly suffered heavy losses from toxic plants would have been less likely to thrive, while those that shared successful observations could pass on small advantages. Sensory clues offered imperfect guidance. Bitterness frequently signaled alkaloids and other defensive compounds. A burning sensation in the mouth or throat, unusual smells, or certain textures could indicate irritation or toxicity. Yet these signals were far from foolproof. Some safe foods tasted bitter, while dangerous ones could occasionally seem mild at first. Our ancestors likely tested small amounts cautiously, watching for effects over hours or days, and shared the outcomes within the group. They also observed other animals. Monkeys stripping fruit from trees or birds feeding on certain berries provided hints worth investigating. Still, animal tolerance did not prove safety for humans. Different species metabolized toxins differently. What one creature could eat with impunity might harm a human child or adult. These observations served as starting points for careful human trials rather than guarantees. Knowledge accumulated slowly through lived experience. It was never written down but lived in demonstrated actions, shared meals, and spoken warnings. Elders and experienced foragers taught the young by example, showing which roots to dig, which leaves to avoid, and how to prepare certain plants. A particular tuber might be safe only after thorough soaking and cooking. A berry could be edible when ripe, but risky when green. These lessons were reinforced through repeated practice, creating cultural traditions that helped communities remember hard-won information. As Homo sapiens and earlier hominins spread across Africa and into new continents, they encountered entirely unfamiliar floras. What had worked in one environment might be useless or deadly in another. Migration presented constant challenges: new leaves, roots, fruits, and seeds that looked promising but whose properties were unknown. Each new landscape required fresh experimentation, careful observation, and the gradual building of local expertise. Archaeological evidence offers glimpses into these practices. Residues on stone tools and dental calculus from Neanderthals and early modern humans reveal consumption of a variety of plants, including tubers, nuts, seeds, and even water lilies in some locations. These findings suggest selective foraging and some level of processing, though the exact methods and decision-making processes remain partly reconstructed from indirect clues. In many regions, knowledge became increasingly sophisticated. Foragers developed practical ways to categorize plants based on appearance, habitat, season, and effects. They learned to recognize dangerous lookalikes, plants that resembled edible species but carried serious risks. Distinguishing the edible wild carrot from the highly toxic hemlock, for instance, could mean the difference between sustenance and tragedy. Such distinctions demanded keen attention to fine details, stem patterns, leaf shapes, smells, and growth conditions. Imagine a foraging party in an unfamiliar valley. They approach a stand of unfamiliar shrubs. One member recalls a similar-looking plant from their previous territory, but notes differences in the bark and the arrangement of leaves. The group discusses, tests cautiously, and decides, sometimes correctly, sometimes at a cost. Over generations, these accumulated observations and shared stories helped communities map their local edible landscapes more confidently. The gradual shift toward agriculture around 12,000 years ago built directly on this existing understanding. Early farmers focused on plants they already knew could be reliable sources of food: wild cereals in the Near East, rice in East Asia, and various tubers and grains in other centers of domestication. Through repeated selection over many generations, humans encouraged desirable traits such as larger seeds, thinner coats, better yields, and in some cases reduced toxicity. Domestication was a long, incremental process of observation and choice. Wild almonds, for example, contain bitter and potentially toxic compounds. Over time, people preferentially propagated trees producing sweeter, safer nuts, gradually shifting the population toward more edible varieties. Cassava, now a dietary cornerstone for hundreds of millions, contains cyanogenic compounds that can release cyanide. Traditional processing methods, grating, soaking, fermenting, and thorough cooking, were developed to make it safe. These techniques likely originated in pre-agricultural experimentation and were refined as cultivation intensified. Cooking and food processing techniques represented major advances. The controlled use of fire, dating back hundreds of thousands of years, allowed people to break down tough fibers, gelatinize starches, and neutralize or reduce many harmful compounds. Soaking leached out water-soluble toxins. Fermentation not only preserved foods but could transform potentially dangerous plants into nutritious staples. Grinding and pounding made roots and seeds more digestible. Drying prevented spoilage, and sometimes reduced toxicity. The invention and spread of pottery further improved these capabilities. Clay vessels enabled better control over boiling, simmering, and steaming. Foods could be cooked more evenly and for longer periods, enhancing both safety and palatability. Other technologies, such as woven baskets for leaching and stone mortars for grinding, expanded the range of plants that could be reliably used. Different cultures developed their own rich traditions of plant knowledge. In many societies, certain individuals held specialized understanding of both edible and medicinal plants. Cultural stories, taboos, and rituals likely helped encode and transmit critical safety information. A tale of a trickster spirit punishing those who ate the wrong mushroom, or a taboo against harvesting certain plants at the wrong moon phase, could serve as memorable carriers of practical wisdom. These traditions were not superstition alone. They were adaptive ways to preserve knowledge in oral cultures. This accumulated wisdom profoundly shaped human history. Reliable plant knowledge supported population growth, more permanent settlements, and the rise of complex societies. It freed time and energy for other pursuits, from tool innovation to cultural expression. Medicinal uses of plants often overlapped with food traditions, creating integrated systems of health and sustenance that varied across continents. Yet the boundary between food, medicine, and poison remained thin and context dependent. When new foods reached unfamiliar populations, such as American crops like potatoes, tomatoes, and maize arriving in Europe and elsewhere after 1492, initial caution was common. People needed time to learn safe preparation methods through experimentation, observation, and exchange with those who already possessed the knowledge. Trade networks carried not only seeds and spices, but also stories and techniques for using them safely. Today, that ancient legacy underpins modern food systems. Supermarkets, breeding programs, and regulations have made plant consumption safer and more abundant than ever, yet echoes of the old challenges remain. We still rely on preparation techniques refined over millennia, and new environmental changes continue to test our understanding. Foraging enthusiasts rediscover the importance of accurate identification, while traditional communities preserve botanical knowledge that science is only beginning to fully appreciate. The next time you eat a simple meal, a salad, a piece of fruit, a bowl of rice, pause for a moment. You are participating in a conversation that began millions of years ago. Every safe bite represents generations of careful observation, experimentation, memory, and sharing. What seems ordinary is actually extraordinary, a living inheritance built through persistence and collective experience. Our ancestors had only their senses and each other. Through patient learning, they turned a world full of potential dangers into sustenance. That hard-won knowledge still feeds every one of us, every single day, and we will never look at our food the same way again.